In the field of gene therapy, we are experiencing a lot of exciting advances. For example, in the last November, the first medication based on the CRISPR-Cas9 got the FDA approval. However, besides this important advancement, there are still many limitations. So one limitation is related to the safety of the treatment. There are still concerns related to the off-target events. And another concern is related to the high cost of this treatment. So we would, in the gene project, we would challenge this issue and provide a new solution that will solve this issue. So first, we should decrease the cost by proposing non-viral vector for the delivery of the CRISPR-Cas9 machinery. Second, we want to make this treatment light switchable, inducible by light, meaning that we can in the future increase the safety profile of this treatment.
Being an IG scientist is a great opportunity to meet new people with new skills, to know more about biological science and the gene therapy, because this is what I would like to know more.
Actually, our tool could be used to treat different kinds of cancer. Because it's safe to be used and we tested it, and even apart from the oncological approach, it could be used even for viral application, such as trying to eradicate infections. Well, even if we couldn't be able to destroy all the damaged cells that we have, in our case, we could actually have an approach by activate again the immune system by targeting some important checkpoints during the cancer cell replication. And by doing this, we could actually do some waking up again our immune system and try to target the cells that we are not able to directly destroy with our tools.
So our hypothesis, what is really happening with this iGene technology, is that we create this nanotransducer. We are targeting the DNA locus using two guide RNAs that will locate the monomers in proximity onto the DNA target. We will have the coupling of the nanorods and the near infrared irradiation. We will have the production of heat that will lead into a thermo-inducible DNA break. And then, biologically, we know that this DNA break now has to be repaired. However, this repair mechanism in cells, they are error-prone. So this can lead into intermutations. And our role here, as my role in the project, was actually to analyze the DNA, testing this nanotransducer in zebrafish, and analyzing the DNA of zebrafish. They were injected with our technology and irradiated in the proper conditions in order to see if actually we have this gene editing. So in fact, the laser was the key changer in this gene editing activation that is light-induced.
The future of therapy for many different kinds of diseases, including cancer, but also other diseases affecting human people, of course, needs to be personalized based on the characteristic of the patient. So, for example, in the case of cancer, the kind of mutation that are inside the cancer to find the best therapeutical options. But on the other side, if we can use, for example, a gene editing tool to target this kind of gene, we should be sure that other sequences in the DNA are not affected. So it is important to take also into account the aspect of safety. So in the case of this hygiene technology, we have this kind of impact in terms of have the capability to modify the on target, but also avoid to modify the off targets. So it is important also from an ethical point of view in curing people.
So let's say that the nanoparticle is the activator of our system, since it allows, by switching on the laser, to generate a heat that is localized on the surface of the plasmon nanoparticle and to induce the double-strand break of the DNA. We have two different cases and two different approaches. In the first one, we have a spherical nanoparticle that is covalently linked with the restriction enzyme and a Cas9. And in this case, when we switch on the laser, we have an increase of the temperature on the surface of the nanoparticle, more or less around 6 degrees, that allow the activation of the activity of the restriction enzyme and the cut of the DNA. In the second case, where we have the dimer, the situation is different because here we don't have a restriction enzyme, we have a couple of nanoparticles, and when the nanoparticles are near one to the other, we have, when we switch on the laser, a really high increase of temperature between the two nanoparticles, around 200 degrees, that allow for the double-strand break of the DNA. The main role in this case of IT in the project about, let's say, the parameter of the laser and the different conditions was to perform the silico analysis, and in order to, let's say, communicate to our partner of the consortium what are the best conditions and parameters of the radiation and modes in order to be able to reach the double-strand break DNA. In the first approach, we found that the best solution is to use spherical nanoparticle of 12 nanometer that allow, when we have a pulse of the laser, and so also a wavelength of 542 nanometer, to reach, as you can see, an increase of 60 degrees in our system. And in the second case, so in this case of the dimer, we found that the best solution is to use not a couple of spherical nanoparticles, but a couple of nanorods that allow us to reach a really high increase of temperature when they are close one respect to the other.
I think this novel strategies is very dynamic because we can use different guide RNA to target different viruses. So in this way, when in the future, probably, but for sure, we have new pandemic situation as SARS-CoV-2 in the last years, we can try to modify the guide RNA after studies the genome of the viruses, the new viruses, and we can target and then cut and destroy this virus.
Well, melanoma lesions are difficult to treat, especially when we are talking about advanced melanoma. We have different types of therapies that work today, but melanoma for us was a challenge to try a different approach of treating cancer. Our idea is to use the gene editing to remove the signature that melanoma use to block the immune system and block the killing of cancer. We have faced many difficulties because melanoma cells are quite resistant to many types of therapies, but the disruption of the genome of these cells by doing hundreds and hundreds of contemporary cuts in their genome, we have to say that there is no way to resist them from this kind of treatment. And hygiene provides a way to localize this huge disruption of genomes just in the melanoma lesion, thanks to the laser technology and our gold nanoparticles.
So I think that the big advantage of the nanotransducer approach is that you can kind of adapt it to different targets. So it's very versatile. You have different building blocks that you can assemble in a customized manner. You have the gold nanoparticle for the delivery and for the photoinducible cleavage, but you can functionalize the gold nanoparticles with a lot of different proteins. And this means that you can adapt our approach to different necessities of different persons and patients.
Okay, so the hygiene technology applies a Boolean logic and a multi-parametric approach in order to have the gene editing. So what does it mean is that the conventional Cas9 approach has only one parameter to be true, that is the DNA targeting, the DNA locus recognition. And after it, the enzyme will actually be able to cut and create the double-strand break that will lead in gene editing. The hygiene technology provides a lot of parameters to be true in order to have the outcome. So it's not only one, the DNA locus recognition, but except of the DNA locus recognition, we have a stimulation that in our case is the irradiation to have a light-activated double-strand break, as well as in our approaches, or the activation of a domain, or in the dimer approach, the coupling of the nanorod. So we have at least 3 parameters to be true in order to have the outcome that is the gene editing. Having this approach now, we are increasing a lot the safety of the system. So in fact, when we have the editing, will only happen in the specific DNA locus that we want to target. And this is how we decrease this effect of off-targeting, that is one of the main limitations why this gene editing is not applied as a gene therapy in clinics.
Molecular surgery, as in an hygiene project, requires a light delivery system. This light delivery system is a really important piece of tooling. We need to control exactly how and where the light is going to be. And for this, we have developed a lab on a chip. This is really a small chip where we control the light, control the liquids. As we said, it consists of 2 components. The light is an integrated photonic circuit. Really, inside this chip, we put the laser light, control it and manipulate it exactly the way we need it to be. As a second part, we have the microfluidics. It's a liquid flow, very small volumes, and we can also control this liquid flow, and the cells are going to exactly the place where we need them to be. There they interact with the light. We enable electrosurgery with the light-induced DNA cutting. This is an important tool to have a reproducible way to do the molecular surgery. Same every time, so we can really build up statistics and really do the safe molecular surgery that is required for the IC project.
So the Prochemia contribution in the IG project is focused on the development of the nanotransducer synthesis. So these ideas are developed in by biologists, by the chemists. They have to convert the ideas into the real structure. And this real structure is based on the modification of the gold nanoparticles functionalized with the polymer layer, and then followed by the attachment of the Cas9 at the surface, which assists in the gene editing process. So The involvement in such a novel and advanced project is really exciting, and we just open new frontiers for gene therapy, and the development of new materials, advanced one, is most challenging and really exciting. So that's why we are really pleased that we are involved in that project.